In this video, we're going to take a look at the process by which white blood cells are able to phagocytize or to engulf and remove materials that don't belong in the body. And then we'll also look at a process that some white blood cells can do that's referred to as extracellular killing, where they destroy something not by bringing it into the cell, but by killing it outside the cell. So here we go. So phagocytosis, that's that process of engulfing and destroying microbes or stuff that doesn't belong in the body. And it requires 4 steps. First, the white blood cell has to find the material to get rid of it. Then it has to adhere to it, so attach it to the membrane. Third, it has to be able to then ingest or engulf the material and bring it inside the cell. And then finally, to destroy it, it must digest it. And there's an illustration in the book. Take a look at this. I strongly encourage you to spend some time checking it out. It is on page 479 in chapter 16. So we're going to pick this apart step by step and take a look at how white blood cells accomplish this process. So the first thing that a white blood cell has to do is find the invader. And it does it through a process that's referred to as chemotaxis. Taxis means orientation toward, or headed in a direction, moving toward. And chemo means chemicals. I hope that makes sense. So chemotaxis is that process of orienting toward or away from a chemical. And so in order to find an invader, phagocytes are attracted to them, so that we see orientation toward a variety of chemicals that are associated with that pathogen or with this material that doesn't belong in the body. So one way that phagocytes are able to find these non-self things, and when I say non-self, I mean a chemical that was made by some other organism than that individual's body. One way that they can find them is through the use of what are called toll-like receptors, or TLRs. These toll-like receptors are found in the membrane of the white blood cell, and they recognize markers that are found in the membrane of the non-self cells, and they're attracted toward them. We see positive chemotaxis, so an orientation toward these markers. The markers can include things like peptidoglycan, which is the material that makes up the cell wall of a bacterium; lipopolysaccharide, another compound that is found in cell walls; flagellin proteins, so these are the proteins that build the flagella or that are associated with the flagella of bacteria; zymosin, and other molecules that may be specific to the pathogen. So any of these materials that are found on the pathogen but not found in the human body can act as markers of non-self cells. So in humans, we know that there are at least 10 of these toll-like receptors found on white blood cells, and each one is particular to some component of the pathogen. So there might be a TLR that's specific to peptidoglycan, another that is specific to lipopolysaccharide, and so on. And just as an aside, they're named toll-like receptors because they resemble proteins that are made by the toll gene. It was discovered in fruit flies, and if that toll gene is defective, then it makes the fly look weird, and toll means weird in German. So a little bit of a contorted source for the name, but that's why it's a toll-like receptor. So there are receptors on the phagocyte, on the white blood cell, that can find molecules, chemicals that are associated with the microbe. Also, microbes and damaged tissue release chemicals that are very attractive to monocytes and to macrophages especially. And so these chemicals will call in a host of monocytes and macrophages to come and help clean up the area. Basophils and mast cells also release histamine This triggers inflammation and also acts as attractants to white blood cells. And finally, chemokines. These are a class of proteins that are referred to as cytokines. These chemokines provide additional attraction for phagocytes. So there are a number of chemicals made either by the self or by the invader, the pathogen that is invaded, that can act as attractants to or for the white blood cells. So the second thing that has to happen in order for phagocytosis to occur is that the white blood cell has to be able to adhere to the microorganism, or have the microorganism adhere to it. You can think of it either way. And so there are receptors that are on the membrane of the microbe that the phagocyte can, has proteins that will attract and bind to. So the capsule, remember the capsule is this gooey layer outside of the cell wall. That capsule can interfere with the ability of the phagocyte to bind, and so encapsulated microbes may be somewhat protected. And so in order for a microbe with a capsule on it to be adhered to, the body must first produce antibodies or other proteins, complement proteins, that will bind to the capsule, and then those become something that the phagocyte can bind to. There's also a protein referred to as the M protein. This is found in the cell wall of microbes that can also interfere with adhering in the same way that the capsule can interfere with adherence. So adhering to the microbe is the next step that must be done, but sometimes additional proteins are required. These are proteins that are made by the host that can adhere to the capsule and then can be used to by the phagocyte to adhere to the microbe. Okay, hopefully all that made sense. The third step that has to happen then is that the white blood cell will ingest the phagocyte. So it engulfs it by making pseudopods. Pseudopods are like, if this is a white blood cell, there's its margin. It can stick out parts of the cell, so the cell can reach out with its cell membrane and engulf some material. So here's the material here, and now it has streamed around it. We call that process of white blood cells being able to move this way cytoplasmic streaming. And so it can stream around the pathogen, surround it, and then bring it into the cell in the form of a vacuole. So now it's streamed around it. It would have kept streaming around it until it then captures it in a vesicle or a vacuole. And now we have it inside the cell in a vacuole that's called a phagosome. So here, this is the phagosome. And here's that bit of microbe. This is some of the extracellular fluid that the white blood cell was crawling through, and it's all captured inside of the cell. And I've got a video for you to look at. Let's see, come on, make it, there we go. So here's a video of a white blood cell that's tracking down, using positive chemotaxis to track down bacteria. So let's take a look at this. I love this video. So you can see these are the bacteria particles. They're the little thing that look like they're diplococci. They're two coccus bacteria. And notice that the white blood cell is streaming, flowing toward it. And it's using positive chemotaxis to find it there. It finally adhered to it. And now it is inside of a vacuole. And I'm going to pause this. Let's see. I'm going to run it back a little bit because I also saw something really interesting earlier when I was watching it. Come on, come back to here. Let's see if I can get it to play. And Nope, I missed it. We're going to go back again, and yeah, I'm trying to see. Oh wait, no, wrong video. I'm sorry, my bad. Okay, so here we are watching it one more time. It is streaming, trying to find it. Getting closer, but still not able to get it. And oh, there it finally adheres, surrounds it, and now it's inside of a phagosome. Okay, so the next thing then that the phagocyte must do is digest the pathogen. And so inside of the cell there are lysosomes. These are small vesicles that are filled with digestive enzymes and chemicals, the defensins, that it uses to well, first it has to fuse this with the phagosome. And once they fuse together, then we refer to it as a phago because it's got the phagocytized material, lyso because it's got the lysis enzymes, some body. So phagolysosomes then are a vesicle inside of the cell that have the enzymes that are going to digest everything. So defensins first perforate the microbe cell membrane. So we need those enzymes that can poke holes in it. And then the enzymes will break up and digest the microbe into smaller molecules that are harmless. And this can take up to 20 minutes to do. And then what we're left with is a material called a residual body. This is just the waste material that cannot be digested, and that can be expelled from the phagocyte. So I've got another video for you to watch of this process. And here it is not going to be a white blood cell we're looking at. This is an amoeba, and it is eating a paramecium. So these are, rather than being things that are inside of our body, these are much larger cells. They're protists that are going to demonstrate this process of digestion for you. So here we go. Come on, I think here we go. There we go. All right, so you can see that we have an amoeba. It is streaming around two paramecia. So the paramecia are now fully engulfed in a phagolysosome or a phagosome. And in a second, you're going to see that, oh, there we are. Now we have the chemicals that have been introduced that are digesting it. And it certainly makes the paramecia concerned about this. And now they've just been digested to goo. And here's what I wanted you to see before. Notice what's happening here. Right there. Oops. Okay, so we're going to replay this. You can see that it is removing. Come on, replay for me, you silly. There we go. We'll see right where that line is pointing to on the amoeba. You'll see that it's ejecting some waste material. Okay, so again, engulfing. Paramecium are like, oh yeah, what's going on? Hey, and oh, now the enzymes are in there. The paramecium are very agitated by that as they are slowly or rapidly digested into just a wad of goo that's inside the, we'll call it a phagolysosome. And so, yeah, there you can see the material, oops, coming out. Dadgummit. I am new to showing videos this way. We're going to zoom, we'll go fast forward. Okay, so pause. So I can't draw directly on the video, but if you look right about where those two lines intersect, you'll see, as I let the video continue playing, you're going to see some extracellular. There's the goo coming out right now. Okay. So that is how the phagocytized material then is digested. So some microbes have developed the ability to defend against being digested. And if they can do this, if they're not digested, they can even multiply inside of this phagolysosome or cause the lysosome to actually destroy the phagocyte rather than destroying themselves, which is crazy to imagine that they can turn the lysosome on the phagocyte. Some of these microbes will form what is referred to as a parasitophorous vacuole, or a PV, that doesn't fuse with the lysosome at all, but instead that vacuole becomes this home for the phagocyte, and it can even use that as like a growing space, as a home base that it can then use to spread throughout the body. This is how Hansen's disease, formerly called leprosy, and tuberculosis operate, and can be safely protected inside the body away from phagocytes because they are inside of cells. As well, the protozoan that's involved with leishmaniasis, that causes leishmania, is able to form these parasitophorous vacuoles as well. And then some microbes produce toxins that can actually kill their phagocyte. Leukocidin, streptolysin, these are some of those enzymes that can do that can actually take out the phagocyte. Okay, so the last bit then in this lecture is about extracellular killing. Hopefully phagocytosis now makes sense. So when a cell does phagocytosis and digestion, that's referred to as intracellular killing, because it's all happening inside of the cell. Extracellular killing occurs as the cell secretes chemicals onto the microbe or onto any dangerous self cells that are found in the body. So perhaps one that has become cancerous or has been virus infected. So eosinophils, they can act as phagocytes, but they can also secrete a toxic enzyme called major basic protein, MBP, that it can use to damage or to actually kill parasitic worms. So if eosinophils can find a parasite worm, then they can attack it and either damage it or even potentially kill it. And then there are a group of lymphocytes called natural killer cells. These NK cells, they look for specific glycoproteins that are on the cell surface. And again, these are cells that are virus infected or have become cancerous. And when they find it, they secrete cytotoxic proteins that perforate the cell and destroy it. So these NK cells are a big piece of how we defend against virus infections and against cancer forming. All right, I think that's the last slide.